Hello everyone, welcome to another lesson from biochemistry. Here we are going to see about the cellular organisms. Welcome to our presentation. So first let's start with smooth endoplasmic reticulum. So what is the smooth endoplasmic reticulum here? This is the rough endoplasmic reticulum because it has the ribosomes. But the smooth endoplasmic reticulum is the one which is smooth because it does not have any ribosomes in it. And it will look like it will be a tubular structure. This is the two-dimensional picture of the smooth endoplasmic reticulum, and this is how it really looks in the three-dimensionally. So it is a tubular structure and it has no ribosomes attached since it is smooth. What is the main function of this smooth endoplasmic reticulum? The main function of it is that it will synthesize steroids and ribosomes. We pretty well know from our previous slides that ribosomes will cause the protein translation. So therefore, this does not have any ribosomes attached to it, and it results in steroids and lipid synthesis. So it is also connected to the nuclear envelope. That also you should be aware of. What is a rough endoplasmic reticulum? Rough endoplasmic reticulum is the one which is seen here, and it has the ribosomes attached to it. What is the main role of ribosomes? The main role of ribosomes is protein synthesis, isn't it? So the main role of rough endoplasmic reticulum is also protein synthesis. But whatever protein that is synthesized in this rough endoplasmic reticulum is mainly used for exocytosis. That protein is sent away and it acts elsewhere. And this rough endoplasmic reticulum, once it synthesizes the proteins, it transports it to the Golgi apparatus. So here is an enlarged view of it. The rough endoplasmic reticulum, once it synthesizes, it sends it to the Golgi apparatus. So for this anterograde transport from the rough endoplasmic reticulum to the Golgi apparatus, we have a COP2 transport. So that is the one which helps in this transport. Similarly, from the Golgi apparatus to the rough endoplasmic reticulum, it is a retrograde transport and that is the COP1 transport. So antigrade is COP2 and the retrograde is COP. What are the free ribosomes? The free ribosomes are nothing but instead of getting attached with this endoplasmic reticulum, it is present freely inside the cell. Its main important function is it produces proteins for intracellular use and it is essential for the cell's internal protein. The proteins which are produced by the free ribosomes are not released elsewhere. It is just used for the protein needs of the cell. Coming on to the Golgi apparatus. A simple way to remember the Golgi apparatus is it is a packing center. So what will basically happen in the packing center? We will write the address, pack the package, and then send it to the necessary area, isn't it? Similarly, the Golgi apparatus also functions in a similar way. It will, once the proteins that are synthesized from the rough endoplasmic reticulum comes to the Golgi apparatus, it will attach a certain glycoproteins or residues and then transport it to elsewhere. So it can transport to the lysosomes or it can exocyte. So the clathrin-coated vesicle transports the protein between Golgi and the endosome. What is an endosome? If the cell wants to intake anything from the exterior, it will form slow buds like this, and finally it forms the endosome, isn't it? So there is this clathrin-coated vesicle. Step one. It's an address kind of this clathrin is. It will transport proteins from the Golgi to the endosome. These clathrin-coated vesicles. Another thing you need to know that this Golgi apparatus is a step which is mainly important in ICEL disease. So let's see what an ICEL disease. ICEL disease is an inherited lysosomal storage disorder. I will tell you in detail about it. What will happen is that there is an enzyme N-acetyl glucosamine 1-phosphotransferase. You can also remember it as NAG1 phosphotransferase. As the name says, it transfers phosphate, isn't it? So, it failure to phosphorylate the mannose residues. Since it does not phosphorylate the mannose residues, so that enzyme is not directed to the lysosome. Instead, that in those enzymes are secreted extra. So the lysosomes become deficient in digestive enzymes. Actually, lysosomes should contain adequate amount of digestive enzymes so that it digests all that unwanted particles which it had engulfed, isn't it? But now, since because of the defect of N-acetyl glucosamine and 1,5-phosphoramidase, there is a failure to phosphorylate the mannose residues. Therefore, instead of getting directed to the lysosomes, they are secreted extracellularly. This results in the buildup of cellular debris inside the lysosomes. So the lysosomes will become big. So normal lysosome is supposed to be very small in size, but here in ICL disease, the lysosomes are enlarged because it does not have the necessary digestive enzymes needed to digest the particles which are present inside the lysosome. So in the USMLE exams, they will ask you, what is that enzyme, in which cellular organelle is that enzyme active? You need to know that the Golgi apparatus is the cellular organelle which will act that. Coming on to this, what is this? This is the mitochondria. We all know that the mitochondria is the powerhouse of the cell. But why is it called the powerhouse of the cell? Because it is that which is responsible for the aerobic respiration. How does aerobic respiration act? Aerobic respiration is nothing but by wherever Krebs cycle and electron transport chain happens, there aerobic respiration happens. So Krebs cycle and electron transport chain occurs in this mitochondria. So because of all that, ATP is being produced. That's why it is called as the powerhouse of the cell. The proton gradient is maintained between the layers of the mitochondria, which helps to produce this ATP. Now, if there is any uncoupling, then the ATP layer is not produced. It's not produced in the normal ratio that it was producing. So, but the oxygen consumption will increase, but the ATP production will decrease. That's what is known as uncoupling and increased oxygen consumption per unit. Coming on to our other next organelle. What are the centrioles and the microtubules? Centrioles, microtubules, mitotic spindles, all these are the organelles related. Centrioles, mitotic spindles. These are the organelles, cellular organelles, which are responsible for separation of the chromosomes and thereby resulting in cell division or replication of a cell or a reproduction. For anything, these are those that are responsible. So we need to know what are those which will inhibit microtubule in cellular. For that, I have a small diagram of it. So mostly the anti-chemotherapeutic agents will act in this case because we don't want the cell to proliferate. If we allow the microtubules to work on its own, it will keep on producing the cells, right? We don't want the cell to proliferate in cancer. Therefore, the microtubule inhibitors are anti-cancer drugs like vinca alkaloids and taxanes like docetaxel, paclitaxel also because they are the microtubule stabilizers. It is also the same way. If the microtubules are stabilized, then it does not shrink. If it does not shrink, the cells are not formed at all. So microtubule stabilizers are the taxanes and microtubule inhibitors are the vinca. And the other one is colchicine, which is an anti-neutrophilic agent because it will inhibit the chemotaxis of the neutrophils because it is an anti-inflammatory. That's how it has to act. So if we have numerous neutrophils being replicated and causing the inflammation, then the disease gets aggravated. Therefore, colchicine will prevent the cell proliferation and therefore its role. And also griseofulvin, it is an antifungal. Also mebendazole, it is an antihelmintic. So all these will also inhibit the cell proliferation. The mechanism of all these are the microtubule inhibitors. Just a small recap. Microtubule inhibitors are anti-cancer drugs such as vincristine, and similarly the paclitaxanes are microtubule stabilizers, colchicine, mebendazole as well as griseofulvin. It is an anti. Let's come to the other important organelle, which is the nucleus. The nucleus is the command center of the protein. Why? Because it is that area of DNA replication and RNA transcription. The DNA will also get replicated here, and from this DNA to form an mRNA, we have it also happens in the nucleus. So, what is the nucleolus? Nucleolus is another organelle which is present inside the nucleus. It will synthesize ribosomal RNA, rRNA. What is a cancer indicator? If the nucleolus is acting in an increased rate, what does it mean? It means that more of ribosomal RNA are produced. The ribosomal RNA are then transported to the ribosome, isn't it? So that means what more of proteins are formed. So when this nucleus cytoplasmic ratio is high, when the nucleus is also increasing and the cytoplasm is also that ratio is high, it will suggest that the nucleus is having an increased activity inside the nucleus, isn't it? So there is an increased mitotic activity inside the nucleus. So high nuclear cytoplasmic ratio suggests that it is a cancer. Another one thing which you need to know for USMLE is that then what about the nuclear envelope breakdown? The nuclear envelope will break down during the prophase phase. How? By means of protein phosphorylation. Once the nuclear envelope protein undergoes phosphorylation, those envelopes will undergo breakage so as to form the microtubules and so that the chromosomes are all separated. So for the nuclear envelope breakdown via protein phosphorylation happens during prophase. So how is this asked in USMLE? They will ask you whether phosphorylation or dephosphorylation causes nuclear envelope breakdown, or what happens during prophase. We need to know that nuclear envelope phosphorylation happens during the. Let's see the other organ. It is the peroxisome. They are the metabolic powerhouse. Right? This is because they will synthesize very long chain fatty acids and also branched chain fatty acids. So both the fatty acid synthesis are happens in the peroxisome. What are the associated disorders? There are some disease, adrenoleukodystrophy, as well as Zellweger disease. All happens in the peroxisome. Let's see about the proteasome. This is another cellular organelle, an interesting organelle which looks like this. This is the proteasome. What is the main action of this? It undergoes protein degradation. How the proteins which are supposed to be degraded undergoes ubiquitination. Ubiquitination will mark the proteins for degradation. Once the proteins have this tag, that is a ubiquitin tag, that is recognized by our proteasome, and it causes the breakdown of the protein. Protein enters inside, it is broken down, and then it comes out into smaller peptides. Those smaller peptides, and finally they are broken down into amino acids. In this way, the amino acids are used for new protein. This is how the proteasome acts. See, this is the protein which are tagged by the ubiquitin and which enters into the proteasome. So once it enters the proteasome, it is broken down into smaller peptides and finally into amino acids. And the ubiquitin are all ready. This is another important thing which might be asked, which is already asked in exams, in US exams. This is such as it is increased in muscle cells during cachexia. When does cachexia happen? Cachexia happens during cancer or in wasting conditions or in starvation. In all these conditions, there is this TNF alpha is getting triggered. Because of TNF alpha, it is that which causes this muscle. So what is that which causes muscle breakdown during cancer and in other diseases is the TNF alpha. So this is also one question. Thank you for listening on this slide. I have an announcement for you. I am starting my mentoring lessons. If you are an USMLE aspirant, or if you want me to, or if you are a medical graduate and if you want me to train you, then here's the good news. I have started my mentoring lessons. Mm, If you like my videos and if you know that there is someone who will be benefited with these videos, kindly share my videos. And if you are interested, you can send me a message in the comments and go ahead and fill the Google form. Looking forward to see you succeed. Have a good day.